The disease cystic fibrosis can come from a mutation in the gene for a specific transporter that is a transport membrane protein. Now, individuals with cystic fibrosis has a mutation that produces a non-functional channel protein whose function is normally to move chloride ions across the cell. So in normal cells, individuals without the mutation pump these ions across the cell membrane, and this leads to a proper balance inside and outside of the cell. Now, normally outside of the cell, we have a layer of sort of a watery mucus, and that's important for normal function, for moving normal products into and out of the cell. But when individuals with cystic fibrosis have this faulty CFTR protein, or this faulty transmembrane regulator protein, those chloride ions can't pass through that channel protein and across the cell. Now, this results in a thick mucus on the outside of that cell, which blocks normal activity and flow of other ions and inhibits other processes in and around that cell membrane. So this is a problem because it affects the function of cells and the organs that those cells are a part of, and then of course the tissues as well. So on the right-hand side, we have a normal lung, and on the right-hand side of the right-hand side, we have a lung of an individual with cystic fibrosis. Now you can see these little pockets that have this mucus lined up inside. Now this causes problems with oxygen transport across those lungs. It can also lead to bacterial infections and also further damage to the lungs because they are not functioning properly. So how do we get from this this proper chloride ion channel to this dysfunctional one? Well, the answer is that it is a difference in the DNA that is coded in our cells. In the process of gene expression, we take the information from our DNA and we use it to make a protein. Now, individuals who do not have cystic fibrosis have the unmutated copy of this DNA, and that DNA is going to be used to make that normal chloride ion channel. Individuals with a change to the DNA produce that dysfunctional chloride ion protein that does not function as a channel. So our DNA is housed in our nucleus, and that sequence for the channel protein is encoded in that DNA in a series of letters, 4 letters, that are read by the cell and used to make a protein. It's kind of like the DNA encodes the recipe for building that ion transporter. So the gene for that CFTR protein, or the normal chloride ion protein channel, is actually copied out of the DNA because the DNA always stays in the nucleus. So we make a copy of the gene that we want to use. And you don't need to know these steps yet, but in a series of steps, that DNA is copied into a short sequence, and then that sequence is taken to the ribosome, whose function you need to know for this chapter, and that ribosome uses that message to build a protein. So the function of the ribosome is protein production. So if this short sequence is encoding our chloride ion transporter, it's going to go to that ribosome where we're going to use it to build our protein. Now, after that, this protein is going to be processed through several other organelles in our cells. You should review the function of those organelles in the cell so that you know which ones are involved in actually helping that ribosome make the final copy of that CFTR protein, which is then embedded in the plasma membrane. Now, if that dysfunctional chloride ion channel is made incorrectly, it may also pop out. So it can block the flow of chloride ions, but it can also pop out depending on the actual structure that is made and the mutation that comes up.